Okay, folks, so I'm going to go ahead and get started into today's information. We're going to finish up our strategies and metabolism. Specifically, we're going to finish talking about oxidized and reduced coenzymes and how they work. After we finish with strategies of the reduced coenzyme or the oxidized and reduced coenzymes, then we're going to get into the citric acid cycle. That is all I actually plan to get through today because the citric acid cycle is going to take quite a bit of time as we talk about each step along the way. Now, as we do talk about each step along the way, when we get into the citric acid cycle, what we're going to be doing is we're going to be looking at the changes that occur in the molecule. We're going to look at the enzyme, and we're going to apply information we learned back in the enzyme chapter to what is going on in the citric acid cycle. We'll also get into ATP-ADP as a metabolic pathway or as a strategy for metabolism, and we'll also get into using these oxidized and reduced coenzymes. So, just as a reminder of what we talked about on Thursday, oxidation is the loss of electrons. What that usually means is it's going to be the loss of hydrogen, or it's going to be the gain of oxygen. Reduction is the gain of electrons. Usually that means that you're gaining hydrogen or you're losing oxygen. With respect to biological systems. Oxidation and reduction must always occur in pairs. So if something is being oxidized, then something else must be reduced. And that's going to be a common theme that we use throughout the process of the citric acid cycle. There is a steady supply of oxidizing and reducing agents that must be available as part of the citric acid cycle. The majority of those are going to be NAD plus and FAD. And then the other two you tend to see in other parts of the process to convert sugars into acetyl-CoA in order to go into the citric acid cycle. So again, if you have something being reduced, the loss of the hydrogens, this is going to wind up, this is oxidation, so the coenzyme has to be reduced. If the coenzyme is oxidized, that means something else has to be reduced. And a lot of times, again, that's going to be through a transfer of hydrogen. So if you'll recall, when we were talking about oxidoreductases, one of the ways we did oxidoreductase was a dehydrogenase, where we removed hydrogen to form a double bond. So again, if something gets oxidized, the coenzyme gets reduced. And it typically gets oxidized through the loss of hydrogen, which is a dehydrogenase, to get to the reduced coenzyme. And then in the electron transport chain, that reduced coenzyme will be reoxidized to form the oxidized form of the coenzyme. And as it does so, what winds up forming in the case of the electron transport chain is that those hydrogens from the reduced coenzymes wind up being donated to oxygen to form water. And so you wind up, that's where the sweat and everything else comes from as our body goes through the cycle. So an example, now, a lot of these things are paired, folks. When we talk about these different pathways, these different strategies of metabolism, you don't have one strategy going on at one time. Back in the enzyme chapter, we would talk about just one reaction undergoing a change and figure out what the coenzyme or the enzyme was. When you're talking about a metabolic pathway, these things occur simultaneously. And so what happens is you show the primary reaction along the in the typical format. In this case, malate going to form oxaloacetate, which is the last step in before the citric acid cycle starts again. But the secondary reaction, which in this case is the reduction of a coenzyme, that is shown as a curved arrow. So you really have two reactions occurring simultaneously. They're paired reactions. Again, remember, that's one of our strategies for metabolism, is to pair reactions so that they have the appropriate amount of energy in order to undergo the process that needs to go. In this case, it's a strategy to pair it with to pair up oxidation and reduction simultaneously. So in this case, what you had to do is you had to look at this malate molecule and look at the oxaloacetate. And when you look at oxaloacetate and malate, you will see that on this carbon right here, what they did is they underwent dehydrogenase or used a dehydrogenase in order to remove hydrogen and make the double bonded oxygen. Because this reaction lost the hydrogens, the NAD+, which is our oxidized form of our coenzyme, it absorbs those hydrogens to make an NADH+. Again, these hydrogens are ultimately going to be used in the electron transport chain to combine with oxygen to form water. Because these reduced coenzymes of NADH have picked up these electrons to form the hydrogen-carbon bond in the nicotinamide, let me get this right, nicotinamide adenine dinucleotide. Because the NADH has picked up those electrons to form a carbon-hydrogen bond, these proton-rich coenzymes, these reduced coenzymes are often referred to as electron carriers. And it's those electrons that we're going to be using later on in the electron transport chain. Any questions so far? I think this is close to where I stopped on Thursday of last week. Okay, so now we're going to get into the citric acid cycle. This is a very generic picture of the citric acid cycle. You have a 2-carbon structure that enters into the citric acid cycle. As we'll find out, this is called acetyl-CoA, where the most important piece is the acetyl. And a lot of times when you hear acetyl, acetone, other things like that, it means 2 carbons. So the 2-carbon enters into the chain. It combines with a 4-carbon compound to make a 6-carbon compound. The 6-carbon compound undergoes isomerization. And then we start getting into the part that's going to ultimately start creating energy. The 6-carbon compound undergoes decarboxylation, I believe it is, where you remove a CO2 and you create a reduced coenzyme at that point.
Did you say they were going
Decarboxylation, I believe it is. Basically, removal of CO2. I believe that's decarboxylation. Now, there's a lot more that's going on here because you also have this reduced coenzyme that's occurring too. So you have a dehydrogenase. So it's actually called a dehydrogenase complex at that point as the enzyme, because you remove a set of, you remove a carbon dioxide, you also remove hydrogen to make the carbon, the 5-carbon chain. This 5-carbon chain undergoes further decarboxylation to remove another CO2, and it also undergoes dehydrogenase to make a reduced coenzyme. At this point, you'll notice you've already made 2 reduced coenzymes and 2 carbon dioxide molecules. This is the location where the citric acid cycle is the location where the carbon dioxide is made in what we're talking about. Now at this point, we have to start building ourselves back up to the original oxaloacetate, which is up here at this C4.
And it goes through a variety of processes to get there. Now, one of those processes actually is an energy producer, which makes GTP. Now, the GTP winds up combined with ADP in order to make ATP. So, when we get into this citric acid cycle in greater detail, you'll see me actually call it ATP instead of GTP. Because again, the GTP, what happens is we combine GTP to make GDP as ADP makes ATP. So almost as quickly as you make your GTP, you make ATP out of the deal, because we combine those two reactions to make an ATP molecule. So it's an energy producing step. As we proceed in trying to reform our oxaloacetate, the next step also is a dehydrogenase. It reduces a coenzyme. It is at this step that all of these coenzymes, this coenzyme, and that coenzyme, are found in the mitochondrial solution, but this particular coenzyme is found in the wall of the mitochondria. And so this one is FAD. So we wind up taking and making FADH2, which is the reduced form of FAD. And again, we continue. And then we have another process which ultimately makes oxaloacetate and another reduced coenzyme. So out of this process, you're going to make 4 reduced coenzymes. 3 of them will be NADHH+, and 1 of them will be FADH2. And ultimately, you'll make a GTP, which quickly converts to ATP out of the citric acid cycle. Now, just so you can see everything going on, I'm going to break it. The next couple of pages in your textbook actually breaks down each step along the way of the citric acid cycle. It is also broken down in the document that you could have printed off of offline. But if you look at page 678 of your textbook, they have a diagram that shows everything that's going on in one fell swoop. So basically, these next 2 pages, what I plan to do today is go over step by step by step to get to the end result. Oops. I didn't do it on this. So let's get into these step-by-step processes. I rattled them off already, but we're going to take our time and go over them. So you start out with oxaloacetate. If we were looking at, let me pull that document back up real quick. Basically, we start right here. This is our 4-carbon initiate into the citric acid cycle. To that, you add an acetyl S-CoA. This The enzyme associated with this is called citrate synthase. And ultimately, what you form is citrate plus HS-CoA. Looking at this, we went from a 4-carbon structure plus a 2-carbon structure to a 6-carbon structure. The enzyme is telling you what occurs. You are synthesizing citrate. And also there's another hydrogen floating around out here, which will go into the hydrogen transport chain. So the synthase enzyme indicates that the small molecule is added across a double bond to make a new molecule. If you look carefully, what we added to was right here. That is where we added across the double bond to make a new molecule.
Will it always be right there where the double bond is, or will it change? Like, or will-
In this particular case, it's where the double bond is. When we get into others, a lot of times, yes, is the answer to your question, because a lot of times it is the double bonds are the most reactive. So a lot of times that's where things can add or move around or change up, is at that location.
Okay.
Now, folks, as far as this is concerned, this molecule comes from digestion. It is the only thing we'll really talk about that comes from digestion, whether that's through the glycolysis chain, or through the lipid decomposition, or through the protein. All we're really going to talk about, because that's all we have time to talk about, is the production of the acetyl-S-CoA, and that comes from digestion. So that's what enters into this ring ultimately. You will notice that besides reduced coenzymes, nothing else nothing else enters this process. Any questions about step number 1?
How would you ask about this on the exam? Like,
I would give you the reaction, I would give you the coenzyme, and I would ask you to explain what's going on.
Okay.
In this case, it is simple. Basically, you are synthesizing citrate from oxaloacetate and acetyl-S-CoA. So now we have our citrate molecule. To that, you add the enzyme aconitase. And you make isocitrate. Now, aconitase isn't, by any of our experiences, something you know. Aconitase is not one of the classes and subclasses that you know. But if you look at this molecule, you could probably guess what it is. Because if you look at this molecule, these two molecules, you'll find they have the exact same chemical formula. Now, this doesn't actually occur in one step. It's a two-step process. There's an intermediate involved. But I'm not going to ask you about the intermediate. What actually happens, I'll tell you about it. What actually happens is you lose water to make the intermediate. And then you gain water to make the new compound. So this is a multi-step process. But what does the aconitase do in this case? Well, if they have the exact same formula, just arranged differently, that means they're isomers. So aconitase is an isomerase. And basically, it just alters the connectivity of the atoms within the molecule. So, thus far, we have combined a C4 molecule, oxaloacetate, with a C2 molecule, which is acetyl-CoA, through a synthase, where we synthesize citrate. The citrate then used the enzyme aconitase in order to isomerize to isocitrate. Again, if I'm asking questions about these particular pieces, I will give you the reaction, I will give you the enzyme. I'm asking you what's going on. So it's recognizing what is going on in the reaction and what the enzyme does to help it all. Now, isocitrate Isocitrate uses an isocitrate dehydrogenase complex. When you see that complex, it means multiple things are going on. It also takes NAD and makes NADH H plus, and ultimately produces alpha ketoglutarate. Plus a CO2 molecule. The first thing I notice is I have a dehydrogenase. Which is an oxidoreductase, which means that oxidation reduction occurs simultaneously. First of all, we removed a CO2. That is where the complex is. In our isocitrate dehydrogenase complex is that removal of a CO2. We also remove two hydrogens to make a double bond. That's where the dehydrogenase comes in. Those two hydrogens that were removed is what makes the reduced coenzyme. So in other words, a lot goes on in this step right here. There's a lot going on in this step right here. You're making a reduced coenzyme because of the presence of the dehydrogenase. So this is the reduced coenzyme. That must mean that alpha ketoglutarate is an oxidized form of the molecule because oxidation and reduction have to go on simultaneously. So again, this is telling a lot of things all in one step. You have isocitrate dehydrogenase, which means remove two hydrogens from isocitrate to form a double bond. Now, this is a complex because not only do you remove the two hydrogens to make a double bond, but also you are removing a CO2 from the molecule. So it's got dehydrogenase and a decarboxylase going on simultaneously. Questions?
So if the coenzyme was oxidized, the product would be reduced?
Correct. If the coenzyme gets oxidized, the product is reduced because they have to go hand in hand.
Gotcha.
Now, this coenzyme is not going to be oxidized until the electron transport chain. But yes, it has to go hand in hand. So if one gets reduced, the other one gets oxidized. Other questions? What am I going to ask you? Tell me the story of what's going on, given the top portion that's in blue. Tell me the story that's going on in this particular step.
We happen to have to know like the citrate and isocitrate.
I'm not asking you to know the names of these compounds. I will give you the names of the compounds. I will give you the formulas. I will give you the coenzyme reaction. I will give you the name of the enzyme. I'm just asking you to tell me what it all means. And yes, I am looking for certain things, such as if the enzyme is reduced, which NADH, H plus is the reduced coenzyme, that means that the isocitrate had to be oxidized to form the alpha ketoglutarate. Because oxidation reduction have to go together. This isocitrate dehydrogenase complex has to have both a dehydrogenase and a decarboxylase in it because of the fact that we removed the CO2. So I'm asking you to tell me the story of what's going on.
Would you ask it in reverse? Like give us the product first and then give us.
No, because citric acid cycle does not move in that direction.
Okay.
It only moves in one direction. That's why it's cyclic. And it always goes from taking oxaloacetate, acetyl-CoA, and ultimately it always reforms oxaloacetate in order to start again. That's what because if you'll remember, we had different pathways that you could take. One of them was cyclic, one of them was spiral, one of them was linear. This is a cyclic pathway and it only goes one direction. Other questions? Okay. And I'm very verbose on the thing that I gave you to help you study for this. So basically, you can read a lot of what I'm saying up here and what I'm verbally saying. Okay. So we now have alpha ketoglutarate. Which is a 5-carbon. This combines with HS-CoA. We have a alpha ketoglutarate. Dehydrogenase. Complex. We have another oxidized form of NAD plus that gets reduced to NADH, H plus, as it goes to make succinyl S-CoA and carbon dioxide. In other words, at this point, we're going from a 5-carbon chain to a 4-carbon chain. Now, in this particular one, the dehydrogenase is kind of more difficult to find because if you look at these structures, you're like, where does the dehydrogenase come in? Where does that removal of hydrogen come in? In this particular case, the removal of hydrogen comes from your acetyl S-CoA. I mean, not acetyl S-CoA, your HS-CoA. That's where the hydrogen comes from that we see in the NADH plus, is from the acetyl S-CoA. Now, again, it's a complex because not only do we remove acetyl S-CoA, but we remove another CO2. Because NAD plus is reduced, that means that the alpha ketoglutarate to succinyl S-CoA is oxidized. Because oxidation reduction have to go together. Can't have one without the other. Later, these H and H plus will wind up going into the electron transport chain and getting combined with oxygen to form water. The only place where we actually add oxygen, so we breathe oxygen, folks, but the only place we use that oxygen in this process with respect to the citric acid cycle and the electron transport chain is in the electron transport chain. That's where the oxygen comes into play. It does not come into play in the citric acid cycle. It's only going to come into play in the electron transport chain. So we've gone from a C5 down to a C4 in making our succinyl S-CoA. Whoops. Okay. In the next part, Succinyl-CoA. Becomes succinate. And again, you lose an HS-CoA, which is going to be important because ultimately we have to use those HS-CoAs to combine with our, to make the acetyl-S-CoA that goes into the process. We need those HS-CoAs to combine with our alpha-ketoglutarate to make succinyl-S-CoA. So we have to continuously be using and producing the HS-CoA in order to continue the cycle. Now this has succinyl-S-CoA synthetase. Now, technically, at this step, what you do is you make GDP and you convert it to GTP. Again, this is an energy producing step, but the GTP rather quickly reacts to form GDP, and ADP reacts to form ATP. So in a sense, it's making an ATP molecule. This is, first of all, this process has to be exergonic in order to drive this process, which is endergonic. Then when it flips around, this becomes exergonic, which drives the formation of ATP, which is endergonic. So what is a synthetase? This one's a very particular one. What is it doing? What is it driving?
Synthesis of succinate.
Not really the synthesis of succinate. I mean, it does drive the synthesis of succinate, but there's something more it does. This one drives phosphorylation. So it drives the addition of phosphate to GDP to make GTP. That's its main goal. That's the main goal in this succinyl-CoA synthetase. Yes, it synthesizes succinate, but more importantly, it drives the phosphorylation of GDP to make GTP. And the ATP is an energy transporter. I know
that is in the way. It just says energy transporter for ATP. Questions? In the next step, gotta make sure I get the right step. Succinate goes to make fumarate. Anybody want to guess what the enzyme is? I know some of you can see it. What am I doing from here to here?
H.
Removing
Yeah.
H2.
Yeah.
So what is that enzyme?
Succinate dehydrogenase.
That's exactly what it is. So this is succinate dehydrogenase. Well, anytime you have a dehydrogenase, that means that you go from a reduced form to an oxidized form. So guess what's going to be going on at the same time? If you go from reduced to oxidized, what has to happen?
You have to add the.
You're on the right track.
Okay, you have to.
You have to have a coenzyme because the coenzyme has to undergo reduction. In this case, it's FADH2. So you have to have something that is reduced something that is oxidized be reduced. They always have to go together, folks. You can't have oxidation if you don't have something else getting reduced. So the entire story here is that the succinase dehydrogenase drives the removal of a set of hydrogens to make a double bond in fumarate. Those two hydrogens are going to get attached to an oxidized coenzyme, in this case FAD, to make FADH2. Again, this step is the FAD comes from the mitochondrial wall. Whereas all of the others, when we play with NAD plus, NAD comes from the mitochondrial solution. FAD is the only one that comes from the mitochondrial wall. So we've taken a C4 and we have kept it a 4 carbon. But what we're starting to really do is we're starting to rearrange that 4 carbon compound in order to get back to our oxaloacetate, which we need.
fumarate then adds water using fumarase to make malate. What type of enzyme is fumarase? That's the only story here. I believe it's a synthase because it synthesizes. And I believe you are correct that it is a hydratase. In fact, I know you are correct because it added water to the molecule. It added the location it added an OH to one carbon and a second hydrogen to the other carbon. So it added water across a double bond, which is a hydratase. So fumarase is a synthase, which combines two molecules. Specifically, it is a hydratase, which adds water across a double bond. Synthase is a generic term. Anytime you synthesize something, you can call it a synthase. But if you want to be more specific, it definitely is a hydratase. So we've gone from C4 to C4. We're still at 4 carbons. It's just now we have an OH group on one of the carbons. Okay, we're going to see if you can figure out the story. I'll add on the test, I'll give you the rest. But I want to know if you can figure out the story, given the fact that we've been talking about this type of thing. So what happened between malate and oxaloacetate? What do you think the enzyme probably is?
The dehydrogenase.
It is malate dehydrogenase. Because you lost 2 hydrogens to make a double bond. Now, where are those hydrogens going to go? The coenzyme. So we're going to also have NAD plus being reduced to form NADH, H plus, and that's where your 2 hydrogens went. So the entire story behind this step is that the malate using malate dehydrogenase loses 2 hydrogens therefore the malate, as it goes to oxaloacetate, goes from a reduced form to an oxidized form. Because oxidation reduction cannot be just one direction. That means somebody else that's in its oxidized form had to be reduced. And what we just did is we went from a C4 to a C4, remaking oxaloacetate so that acetyl-S-CoA can recombine with oxaloacetate and start the cycle again. So, again, you're going to make 2 CO2 molecules. You are going to make 4 reduced coenzymes. 3 will be NADH plus, or NADHH plus, and 1 will be FADH2. And then you will make a GTP-ATP molecule as part of one cycle of the citric acid cycle. Now think about that. Glucose has 6 carbons. And what enters this is 2 carbons. So one glucose molecule, if it has 6 carbons and has to be down to a 2 carbon structure to enter, that means for one glucose molecule, you make 3 acetyl-S-CoAs. If you make 3 acetyl-S-CoAs, that's C2, that means you're going to perform the citric acid cycle 3 times for one glucose molecule. So for one glucose molecule, technically you're going to make 6 CO2s. You're going to make 3 GTP-ATP. You're going to make 9 NADHH pluses and 3 FADH2s. That's 12 reduced coenzymes that are going to go into the electron transport chain. And as we'll see, the electron transport chain then takes all those H pluses and converts them right back into, converts that into energy molecules of ATP. So it drives the formation of our ATP molecule. Now, we talked about, and when we were talking about enzymes, we are talking about the fact that enzymes have to be controlled. If you have an extensive amount of ATP, for instance, then we don't need this citric acid cycle to occur. And so the ATP itself and the ADP become regulators. If you have too much ATP, it's going to regulate how much, whether the citric acid cycle can occur. Just like if you have more ADP, you want the citric acid cycle to occur because you want to convert it into ATP for energy. And so these molecules that we make, the reduced coenzymes and the ATP, ADP, those are regulators for the citric acid cycle. They basically turn the cycle on or off. Depending upon what needs to be done in order to keep the cycle, in order for us to have the right amount of energy within our bodies.
questions about today? So again, with the citric acid cycle, what are you gonna have to know? You're gonna have to be able to explain each step. I'll only give you 2 or 3 of them. And you're going to have to give a written explanation of what's going on. I'm not gonna give you all of them. That took me an entire class period. But I'll give you 2 or 3. And you're gonna have to give me a written explanation as to what's going on with each step. You will receive the reactants. You will receive the enzyme. You will know whether or not there is a coenzyme associated with it, and you'll receive the products so that you can explain what's occurring. You need to be aware of what starts the citric acid cycle. It's acetyl-S-CoA combining with oxaloacetate. If you don't have acetyl-S-CoA, you're not going to start the citric acid cycle. You need to know what one cycle produces. This is a great picture of that. That particular picture, I want to say, is found on page 675, figure 21.7. And you need to understand the top paragraph on page 679, which basically is the rate of the citric acid cycle is controlled by the body's cellular need for ATP and reduced coenzymes for energy. For example, when energy is being used at a high rate, exercising, eating, whatever, ADP accumulates and acts as an activator for the isocitrate dehydrogenase. That is your first that is the coenzyme. I'm sorry, not the coenzyme. That is the enzyme that goes from this step to this step.
What page number was it for that paragraph?
679.
679.
When the body's supply of energy is abundant, meaning you've got plenty of ATP and plenty of NADH and plenty of FADH2. Those ATP and NADH actually act as an inhibitor for those enzymes. And so it tells it to stop. So it's not really understanding what is the inhibitor and what is the activator. It's understanding the concept of an activator and an inhibitor. So I'm not asking you to know that ADP and NADH is the inhibitor. I'm asking you to know that if you have an excess of ATP and an excess of NADH+, you don't need any more. So the enzymes are inhibited so that the citric acid cycle doesn't continue. But if you have an excess of ADP and an excess of oxidized coenzymes, then the reaction needs to occur. And so you have the enzyme is activated so that the reaction will occur and the citric acid cycle occurs. It's not knowing the specifics, it's knowing the concept of, again, inhibition compared to activation and why we would do them. Questions?
So with that, just to clarify, you're saying if there's too much of the reduced coenzymes, it won't continue the cycle? Or is it too much of the oxidized?
No, if it's too much of the reduced,
Oh, okay. And it's like, there's no need for more.
because if there, right, if there is too much of the reduced, there's no need to, hold on. If you have an excess of ATP, so if you have an excess of reduced coenzymes, you're also going to have an excess of ATP. Because for every one cycle, you make an ATP. So if you have an excess of ATP, you also have an excess of reduced coenzymes. So it's going to stop the cycle. Whereas if you have the reverse of these, so oxidized and GDP or ADP, then it starts the cycle. Does that make sense? Okay, so Thursday is our last class. It's hard to believe. And yet, it's not. I know, it's both. It feels like the semester just began. And yet, everybody is ready. What we're going to do on Thursday is we're going to talk about the electron transport chain. That is our last topic, and that is all I'm going to go over on Thursday is the electron transport chain. The electron transport chain is going to be the location where all these reduced coenzymes lose their protons, lose their electrons, to become reactivated, to become oxidized, and where additional ATP is formed from those electrons and phosphorylation. So we're going to talk about the electron transport chain, and that will be the end of the semester. I will also probably on Thursday give you a little insight into the test because I actually have it written for a change early. So I will try to give you a little bit of insight into the exam. To help out in preparation for the last exam. Any questions for me? Yes.
Last Thursday, you mentioned the columns on Blackboard, so we could figure out what our grade would be.
Okay.
Are those on there yet?
Not yet. They will be up probably by Thursday because one of my goals is to finish grading your labs. And as soon as I get your labs graded and placed into Blackboard, then I can release those columns because, and also as soon as your quiz is done for today. So I don't want to release it until all of the information is there, so that it gives you the most accurate picture possible. Otherwise, it just confuses people because they have to say, well, I still have a quiz, I still have labs, what's my grade? And I found it just confuses people more. So I would rather wait until everything's in. I also need to add bonus, and don't forget there is a bonus opportunity tonight. Again, it's what it is if you come to the video talk, which you had to sign up for. Did I give you all that information? Okay. You sign up for it, you come to the talk, just being there is 5 points. If you take notes and turn them in on Thursday, that is an additional 5 points for 10 points, for an additional 10 points. Plus I've got to add all the cans upstairs, all the food upstairs into those points. But that's another thing I have to do before the column is officially up to date. So I'm working on it. It's there, it's just hidden. Other questions for me? Well, that's all I have for today then.